The body's immune system is highly effective at detecting diseased cells, but this is often not the case in cancer. All cells, including cancer cells, have molecules on their surface that present peptides known as antigens to T cells of the immune system. Most antigens don't cause an immune reaction. However, in cancer, antigens specific to the cancer, referred to as neoantigens, can be recognized as foreign by T cells. Cancers that have lower levels of neoantigens aren't cleared by the immune system and do not significantly respond to current immuno-oncology therapy. Greywolf aims to address this limitation by changing how antigens and neoantigens are made to increase cancer visibility. Normal proteins and proteins mutated in cancer are broken down by the proteasome into peptides and transported into the endoplasmic reticulum, where a proportion are processed by the protease ERAP1 before binding to MHC class I. Antigens and neoantigens bound to MHC class I are then transported to the surface and presented to T cells. To increase cancer visibility, Greywolf is modulating ERAP1 activity. ERAP1 is a protease that trims certain peptides to create antigens and neoantigens of the optimal length for binding to MHC class I. ERAP1 overtrims some neoantigens, preventing their binding to MHC class I. Modulation of ERAP1 with a drug will prevent the overtrimming of these neoantigens, allowing them to bind MHC class I, leading to an increase in neoantigen presentation. Modulating ERAP1's activity will also change the pool of neoantigens able to compete for binding to MHC class I. As a result, neoantigens that are not processed by ERAP1 and do not normally bind MHC class I are now presented. The result? ERAP1 modulation causes new neoantigens to be displayed and other neoantigens to increase in number. This enables the immune system to detect and destroy a now more visible tumour.